that's something. Okay, so we have two talks from graduate students today. The first is Sheila Walsh, molecular biology PhD student in the Wall Lab, to tell us about Myxo social interactions. Yeah.
Thank you so much for the introduction. I'm very excited to be here and tell you about what I've been working on for the past few months in the Wall Lab. Let's go ahead and get started. So some Myxococcus xanthus basics is Myxo is a gram-negative bacteria, which means it has an outer membrane and a cytoplasmic membrane with a thin layer of peptidoglycan here in the middle. It has two types of motility used by two different motors. First is S motility, shown here, where it is able to move as a social group, as well as its adventurous motility, where it moves as an individual cell. Another very important aspect of Myxo is that it is soil dwelling. It is estimated that a gram of soil can contain thousands, if not tens of thousands, of species of different bacteria. So in this microbial jungle, it is really important that Myxo needs to be able to recognize who is who. So Myxo have very complex social lifestyles that can be broken down into two different parts, notated here with this yellow line. Its interactions with other bacteria and its interactions with itself. Today, we'll be focusing the first part of the talk on predation and kin discrimination. So let's get started. So part one is talking about how family feuds led to the discovery of new functions. First, we're going to go through a quick background. We'll talk about objectives and how they lead into the workflow, the results, conclusions, and future directions, and then we'll get into part two of the talk. So a long, long time ago, in 2003, in a land far, far away, Germany, the Belliser group took 100 soil samples from a 16 by 16 centimeter plot. Of these soil samples, some did not produce Myxo, notated here in the white, where others did, and a total of 78 individual Myxo isolates were extracted. If they had more than one isolate in that sample, only one was kept. Of these 78 isolates, there equal total a total of greater than 50 social compatibility types, which were tested here, showing the colony merger incompatibility. You can see the incompatibility by this little demarcation line where the bacteria have not merged with each other. Here you can see that there is compatibility. They have merged. There is no line. This moving forward, we can talk a little bit more about how Myxo is able to mediate this these incompatibilities. First is a more general amongst bacteria. More general amongst bacteria is the type 6 secretion system. It is in about 25% of gram-negative bacteria. I will liken this to a contractile sphere that is able to extend and inject an infector into a neighboring cell. The outer membrane exchange is a little bit more Myxo-specific, where two cells will join together using the TRA cell surface receptor. They bind homotypically, and during this binding, the membranes fuse. When they fuse, the membrane is exchanged as well as parts of the membrane, which would be our lipids, proteins, as well as toxins. If the recipient cells do not have the immunity to the toxins, cell lysis and death occurs. And if they do have immunity to the toxin, then we see harmony in this green. So flash forward to work that our group has done. We took two clades from the samples and wanted to look about more into these social incompatibilities. So we've already talked about the type 6 secretion system as well as outer membrane exchange. So when those two were knocked out in the bacteria, we'll look at this blue clade right here. We see that most of the antagonism has gone down, showing here in green. However, you will notice that there are still some red squares showing that there is continued antagonism. I would also like to point out here that these squares down here notate that we do not have these strains created and we have not studied them. So what the heck is still causing all of this antagonism? So what we notice when we sequence their genomes is that they are 99.9% identical except for genomic islands which harbor these RHS proteins and toxins. So for two of the isolates, 7 and 64, we went ahead and knocked out their RHS toxin or their protein. And we take a look now back at resolving the social compatibilities, and we see now the antagonism has been resolved. There is harmony. Now that we've had a little introduction to the RHS proteins, we'll move into my specific protein of study, which is RHS2. The reason why we chose this one is that it is very similar to what we have in our lab strain 1622. So traditional RHS proteins are usually small polymorphic toxins that are associated with the type 6 secretion system. They're found in many gram-negative bacteria, and they contain this dipeptide repeat. That's how it gets its name. However, this RHS2 is a very large polymorphic toxin over the 3,000 amino acids, and it's actually associated with the SET pathway. So my objectives were to compare these RHS2 structures with additional RHS2 in Myxo as well as in other bacteria using bioinformatics and compare the predation of wild type versus knockout strain. So here's a little bit about the workflow of what I've done. We created a 6679 or RHS2 knockout, and we're going to perform some experiments on plates, as well as using, again, bioinformatics to compare the RHS of proteins. So starting off with bioinformatics or some literature search, we have notated that 6679 has been linked to motility previously, as well as it has been linked to predation by the Kirby group, and it has many polymorphic homologs and orthologs. Here we can see the crystal structure of Vibrio parahemolyticus. It's RHSP protein, and we can notate that this structure on the outside is a cage that is harboring this toxin shown here in red. Additionally, if we look at RHS9 found in Myxo, we notice the same type of structure, or a cage that could hold a toxin. Here we have the alpha fold and pi mole rendering of our Myxo, or of our RHS2, and hopefully I can get the video to play for you. So this, since it's a very large amino acid, alpha fold doesn't really like to do more than about 1,500 amino acids. So I'll orient you. Here, this goldish or orange color is the first 1,500 amino acids of the protein, and this greenish color is the second half. We don't believe that this is the true structure, although it's very close to it, because we don't have the toxin inside of this cage-like structure. So moving on to the predation assays. In this assay, what I have done is I have taken E. coli cells and I have concentrated them down and plated them onto a minimal media auger, which will kind of force predation. Here at 24 hours, and then after that, I spotted whether it was the knockout strain or the wild type strain. Here at 24 hours, we can see a clear difference between the clearing of the E. coli compared to this wild type. At 48 hours, there is not much difference, but you can see the phase variation here in the wild type. A different type of predation assay we have done, or I've done, is taking different concentrations of E. coli cells. So for every one Myxo, there is 25 E. coli and greater concentrations as we go here to the right. Again, at 24 hours, we can see the most difference between wild type and the knockout. I would like to note that even here at 1 to 100, the knockout strain seems to be doing better than our wild type, or roughly about the same as our wild type at the 24-hour mark. Again, at 48 hours, We do see some differences, but they're not as vast. So in conclusions, we do see that there are similarity in structures between RHS in Myxo, RHS out of Myxo, and my particular RHS2. We also note that the knockout strain seems to heal quicker than the wild type. This could be because it's not involved in kid discrimination any longer. Now it is able to focus its resources and energy on predation rather than worrying about its kin. So some future directions I have is to optimize the antibodies we have against this RHS2. We have created overexpressing strains, and because I haven't got this to work, I wasn't able to show the experiments with overexpressing strains. We'd like to determine its role in kin discrimination because we don't know of it quite yet, as well as compare predation of other lab strains that we have. Since there have been, it has been cited in motility, we'd like to also compare the motility of knockout, overexpressor, and wild type strains. And then explore its mechanism of action, because we're not sure exactly how it's working. So the second part of my talk, which I'm going to call Friendsgiving, the sharing between auxotropes, we're going to look at a different interaction with Myxo, this cross-feeding. So this one is going to be somewhat similar. We're going to go over background, objective of workflow, results, conclusions, and future directions. Some thanks, and then we'll get on to my amazing friend Martin's talk. So a little bit about background of cross-feeding. Cross-feeding occurs throughout microbial habitats, including our gut and in the soil, and it can occur with the same species or amongst other species of bacteria. We know that Myxo utilize their sibling cooperation for this outer membrane exchange, creating fruitibodies with clone mates, but does sibling cooperation also rescue auxotropes? So previous study in our lab, we looked at auxotropes with the prototrope, and we noticed that when the auxotrope and prototrope were plated together in minimal media containing additional calcium, that the growth of the auxotrope is significantly decreased. In another experiment where they're looking at different auxotropes, we see that again in this minimal media that contains calcium, the competitive index, or the ability for the wild type to kill the auxotrope, is increased. It's shown here. So we found that this is dependent on type 6, killing the type 6 secretion system. Since the auxotrope is starving, it is unable to make the immunity for the toxin for the type 6 secretion system, and therefore it ends up dead. So my objective was to test whether these auxotropes ability to share with each other. So we plated them pairwise to see who would help who. Out of all of these different pairings, only 2 really showed some promise. So here we're looking at the 72-hour mark, where we can see that there is modest growth or light growth compared to the auxotropes alone when they're mixed together for both threonine and tryptophan, as well as with histidine and threonine. Obviously, it is modest growth because if we look at wild type at 72 hours, you can see a huge difference. So conclusion for this one, although there is modest growth of the auxotropes, we don't know if it's actually occurring from the cross-feeding. So in future directions, we'd like to develop a more sensitive assay. There is a possibility that the outer membrane exchange is what is allowing the growth of these. So what we could do is knock out the TRA receptor in one of the strains and see if this growth still occurs. Another option we have is to develop new strains. Previously, the auxotropes that we have an antibiotic marker for CAN resistance, so we could create new strains with TET resistance and look at how many colony-forming units come from both. So I would like to thank my current lab members, previous lab members who, without their work, I wouldn't be here talking to you today. And thank you so much for Vincent for showing me how to utilize AlphaFold and Chymol so we could get that really pretty video. Thank you to my funding sources, INBRI and the NIH. And if you want to get a more up-to-date recall of what my research is, follow me on Instagram, Myxo on my mind. With that, I will gladly take any questions. Questions, concerns, emotional outbursts. Yes,
How good were the AlphaFold simulations? Like, what was your PLDT?
that is a great question. I do not have that data, but I can definitely get it to you. This is the rank number 1. I'm not really sure, but since it does show similarity in structure, I'm assuming it's okay. Great. Yes, sir.
So you were talking about the toxin, so your RHS
Mhm.
forms like this beta-oral structure that looks like a bunker
Yeah.
hollow inside, and
Yeah.
then you were saying something about toxin.
So what we think is that in these proteins, they're known toxin domains. And so what we think is that the toxin is being shuttled around inside of this cage, allowing it to be shielded from the cell itself if there is toxicity, and that it is transferred now to the secretory pathway for the type 2 secretions.
So that like the toxin is per se the RH2.
We think so, yeah.
Okay, so the pointing thing. Okay.
Yeah.
Yeah.
It's hard. I want to, I don't know how I can do it, but I would like to maybe chop it up into different domains and see if that changes the structure in AlphaFold, because it's really hard to get the whole thing in there. I don't think AlphaFold knows where to do any cleavage if cleavage is happening.
But do you know which part of the protein is the one that has the toxicity?
Yeah, yeah. So this one is the first, and I believe that it shows that technically it shows in the RHS2. Let me see if I can come back to that. In this RHS2, this is technically supposed to be the toxin here in this one, but this is different than what we see in our 1622 strain. So this is in the environmental isolates, which is really weird because if we go to this is the second half. So on here, there is this little section that this could be the toxin, this little green part that comes out here could be the toxin, but we're not sure quite yet. Dr. Gonowski?
Could you please help me understand why you think that this mixing of auxotropes could be helped by outer membrane exchange? What could possibly be the mechanism?
So what we think could happen is during outer membrane exchange, we have lipids and proteins being exchanged. So possibility there could be amino acids also being exchanged during that part. It's been shown, I think, in other papers. I don't have the resources to show that with you right now, but in previous studies, we see that it's moving a whole bunch of other stuff. So why couldn't it move the amino acids as well?
That would imply that there are a bunch of amino acids just floating in the territory. Is that so?
I don't have a good answer for you. So that's why if we check it, I mean, it's a possibility. It's a possibility. I don't know exactly what would be exchanged during that, but a good way to try would be to knock out that receptor and see if we do see the growth.
Other questions? Okay, well, let's actually got that.
And you're different. I know, yeah, I would do that.